Hi everyone, welcome back to my channel. In this video, we will learn about gram-positive and gram-negative cell walls. Gram-positive bacteria normally have cell walls that are thick and composed primarily of peptidoglycan. Peptidoglycan in gram-positive bacteria often contains a peptide interbridge. In addition, gram-positive cell walls usually contain large amounts of teichoic acids, that is, the polymers of glycerol or ribitol joined by phosphate groups. Amino acids such as D-alanine or sugars like glucose are attached to the glycerol and ribitol groups. The teichoic acids are covalently connected to either the peptidoglycan itself or to plasma membrane lipids. In the latter case, they are called lipoteichoic acids. Teichoic acids appear to extend to the surface of the peptidoglycan, and because they are negatively charged, help give the gram-positive cell wall its negative charge. The function of these molecules are still unclear, but they may be important in maintaining the structure of the wall. Teichoic acids are not present in gram-negative bacteria. The periplasmic space of gram-positive bacteria, when observed, lies between the plasma membrane and the cell wall and is smaller than that of gram-negative bacteria. Even if gram-positive bacteria lack a discrete, obvious periplasmic space, they may have periplasm. The periplasm has relatively few proteins. This is probably because the peptidoglycan sac is porous and any protein secreted by the cell usually pass through it. Enzymes secreted by gram-positive bacteria are called exoenzymes. They often serve to degrade polymeric nutrients that would otherwise be too large for transport across the plasma membrane. Those proteins that remain in the periplasmic space are usually attached to the plasma membrane. Staphylococci and most other gram-positive bacteria have a layer of proteins on the surface of their cell wall peptidoglycan. These proteins are involved in the interactions of the cell wall with its environment. Some are non-covalently attached by binding to the peptidoglycan, teichoic acids, or other receptors. For example, the S-layer proteins bind non-covalently to polymers scattered throughout the wall. Enzymes involved in peptidoglycan synthesis and turnover also seem to interact non-covalently with the cell wall. Other surface proteins are covalently attached to the peptidoglycan. Many covalently attached proteins, such as the M protein of pathogenic streptococci, have roles in virulence, such as aiding in adhesion to host tissues and interfering with the host defenses. In staphylococci, these surface proteins are covalently joined to the pentaglycine bridge of the cell wall peptidoglycan. An enzyme called sortase catalyzes the attachment of these surface proteins to the gram-positive peptidoglycan. Sortases are attached to the plasma membrane of the bacterial cell. Coming to the gram-negative cell walls. Gram-negative cell walls are much more complex than gram-positive walls. The thin peptidoglycan layer next to the plasma membrane and bounded on either side by the periplasmic space may constitute not more than 5 to 10% of the wall weight. In Escherichia coli, it is about 2 nanometer thick and contains only 1 or 2 sheets of peptidoglycan. Some periplasmic proteins participate in nutrient acquisition, for example, hydrolytic enzymes and transport proteins. Some periplasmic proteins are involved in energy conservation, for example, the denitrifying bacteria which convert nitrate to nitrogen gas, and bacteria that use inorganic molecules as energy sources have electron transport proteins in their periplasm. Other periplasmic proteins are involved in peptidoglycan synthesis and the modification of toxic compounds that could harm the cell. The outer membrane lies outside the thin peptidoglycan layer and is linked to the cell in two ways. The first is by Brown's lipoprotein, the most abundant protein in the outer membrane. This small lipoprotein is covalently joined to the underlying peptidoglycan and is embedded in the outer membrane by its hydrophobic end. The outer membrane and peptidoglycan are so firmly linked by this lipoprotein that they can be isolated as one unit. The second linking mechanism involves the many adhesion sites joining the outer membrane and the plasma membrane. The two membranes appears to be in direct contact at these sites. In Escherichia coli, 20 to 100 nanometer areas of contact between the two membranes can be seen. Adhesion sites may be regions of direct contact or possibly true membrane fusions. Lipopolysaccharides, these are large complex molecules contain both lipid and carbohydrate and consist of three parts, that is lipid A, the second one is core polysaccharide and third one is O side chain. The lipopolysaccharides from Salmonella has been studied most and its general structure is described here. The lipid A region contains two glucosamine sugar derivatives, each with three fatty acids and phosphate or pyrophosphate attached. The fatty acids attach the lipid A to the outer membrane, while remainder of the lipopolysaccharide molecule projects from the surface. The core polysaccharide is joined to the lipid A. In Salmonella, it is constructed of 10 sugars, many of them unusual in structure. The O side chain or O antichain is a polysaccharide chain extending outward from the core. It has several peculiar sugars and varies in composition between bacterial strains. Lipopolysaccharide has many important functions because the core polysaccharide usually contains charged sugar and phosphate. Lipopolysaccharide contributes to the negative charge on the bacterial surface. Lipid A also helps stabilize outer membrane structure. Lipopolysaccharide may contribute to bacterial attachment to surfaces and biofilm formation. A major function of lipopolysaccharide is that it aids in creating a permeability barrier. Lipopolysaccharide also plays a role in protecting pathogenic gram-negative bacteria from host defenses. The O side chain of lipopolysaccharide is also called the O antigen because it elicits an immune responses. This response involves the production of antibodies that bind the strain-specific forms of lipopolysaccharide that elicited the responses. Importantly, the lipid A portion of lipopolysaccharide often is toxic. As a result, the lipopolysaccharide can act as an endotoxin and cause some of the symptoms that arise in gram-negative bacterial infections. If the bacterium enters the bloodstream, lipopolysaccharide endotoxin can cause a form of septic shock for which there is no direct treatment. The outer membrane is more permeable than the plasma membrane and permits the passage of small molecules like glucose and other monosaccharides. This is due to the presence of porine proteins. Most porine proteins cluster together to form a trimer in the outer membrane. Each porine protein spans the outer membrane and is more or less tube-shaped. Its narrow channel allows passage of molecules smaller than about 600 to 700 Daltons. However, larger molecules such as vitamin B12 also cross the outer membrane. Such large molecules do not pass through porins.